Assalamu alaikum, Psychosis Exceptionalis. This is the third video in the antibiotics course. In the previous video, we took the cell and its types, eukaryotic and prokaryotic cells, and the main differences between them. In this video, we will be focusing on the prokaryotic cell, particularly the bacterium and its classification. Let's get started.
فأنا طالع على الباب إلى زوجتي بتنادي علي. استنى عدنان، جبت لي الوقتين صف هيك جابت كاميرا تك 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 صورتني. حسيت إنها بتودعني. وفعلا كنت طالع على الموت. إسعاف تعرض للقصف عدة مرات في أماكن كنا نتجنبها بسبب القناصة. الطبيب الفلسطيني، الجراح الفلسطيني اللي خاض الغمار، كثير من جراحينا استشهدوا وهم بيأدوا عملهم.
When bacteria are classified, there are many considerations. First, gram stain. Are they gram positive, gram negative, or variable, atypical, or just not staining? Morphology. Are they cocci, bacilli, cucubacilli, or spirochetes? Serotype for streptococci and enterococci. Are they group A, group B, group D streptococcus? And by the way, the group D streptococcus is an old classification for this group, and it is now classified as a separate group called enterococci. Biochemical reactions. Are they lactose fermenting, non-lactose fermenting, catalase positive, catalase negative, and so on? Growth requirements. Do they need oxygen or not? What are the differences between gram positive and gram negative structurally? Gram positive have much thicker cell wall, peptidoglycans. The glycans or the polysaccharides that are cross-linked by peptide chains, peptidoglycans. Whereas the gram negative have much thinner cell wall. The gram positive have teichoic acid, which is inserted in the cell wall, whereas the lipoteichoic acid is inserted in the cell membrane. The lipoteichoic acid stimulates the release of tumor necrotic alpha, interleukin 1, interleukin 6, which are responsible for inflammation and fever. Both cells have cell membrane, which is composed of bilayer phospholipids that contains many different proteins. The most important one for us, the penicillin binding protein or the transcriptase enzyme, which is responsible for the last step in the cell wall synthesis of bacteria. And it is the target for beta-lactam antibiotics such as penicillins. Unlike gram positive, gram negative have an outer membrane which is composed of an outer layer and an inner layer. The outer layer, it contains lipopolysaccharides or endotoxins, which is secreted from the dying bacteria, and composed of lipid A, which is pyrogenic. Pyro, temperature. Genic, synthesis, the synthesis of temperature. Fever, because it stimulates release of tumor necrotic alpha, interleukin 1, interleukin 6, and all of these cytokines are responsible for fever and inflammation. All antigenic, recognized by antibodies. The inner layer of the outer membrane contains several proteins such as porin, protein, which is a selective transport through the outer membrane for molecules including the antibiotics. So, beta lactam antibiotics, which work on penicillin binding proteins, need to pass through this porin to be effective in gram negative bacteria. Gram negative also have periplasmic space, which is found between the outer and the inner or cytoplasmic membrane, and it contains the thinner peptidoglycans and other proteins or enzymes such as beta lactamase. The beta lactamase is responsible for the beta lactam antibiotics resistance. Now we have a question. Why penicillins antibiotics more effective on gram-positive bacteria than gram-negative bacteria? Even though gram-positive have more or much thicker cell wall peptidoglycans, the answer has two concepts. First, it doesn't matter if the cell wall is thick or thin, because the cell wall is highly porous and the molecules, including the antibiotics, can pass through it easily to target the penicillin binding protein. Whereas in gram negative, they have an outer membrane which is selective bilayer phospholipid, so the antibiotics need to pass through the porin transport to target their penicillin binding protein. Second, if the gram positive have beta lactamase enzymes, they are secreted and released outside the cell to target the beta lactam rings of the antibiotics, even though far away from the cell. It seems good for the bacteria. It is not actually, because in gram negative, they have an outer membrane which prevents the releasing of beta lactamase outside the cells. So they are highly concentrated in the periplasmic space. There are tons and tons of beta lactamase, as you can see here, waiting for their enemies, the penicillins. When the penicillins or the antibiotics pass through the porin into the periplasmic space, they will be destroyed immediately by the beta lactamase. That's why gram-negative bacteria are more resistant against beta-lactam antibiotics, particularly the penicillins. Both bacteria have a capsule, which is a polysaccharide structure, and it is a virulent factor against phagocytosis. They also have flagella that help them in their movement either toward the nutrients or far away from their enemies. They also have a pilus or a sex pilus, which is responsible for the conjugation process. Conjugation is a process in which a bacterium directly transfers genetic material to another bacterium via a pilus. The genetic material, of course, is the plasmid DNA and not the chromosomal DNA, because bacteria cannot transfer chromosomal DNA while they're still alive. Let's imagine that the plasmid DNA contains the genes responsible for encoding beta lactamase enzyme. So the F positive bacterium is resistance against the beta lactamase sensitive antibiotics as penicillin G. Whereas the F negative bacterium, which doesn't have the plasmid DNA and the genes responsible for encoding the beta lactamase enzyme, so it doesn't have the beta lactamase enzyme and it is sensitive to the antibiotic. By the six pilus connects between the two cells, and in this direction from the F positive to F negative, the conjugation process starts, and the plasmid DNA is transferred from F positive bacterium by the sex pilus to F negative bacterium by the conjugation process. As a result of that, the F negative bacterium turns into F positive by having the plasmid DNA. So it is now beta lactamase sensitive penicillins resistant bacteria. The gram stain is the most widely staining method in microbiology, and it is a differential stain because it divides the bacteria into gram positive and gram negative. What are the steps of gram stain? Start adding your sample, and the sample may be urine, blood, saliva, cerebrospinal fluid, or any other sample that contains bacteria to a glass slide. And it must be fresh because um, when the cells get old, they start losing their ability to retain the stain. After that, apply the crystal violet, the most important part in gram stain. But before that, we should heat fix the smear of bacterial culture by passing and moving the glass slide above the flame of Bunsen burner to make it adhered and to allow the sample to more readily take up the stain. So add the crystal violet to the previously heat fixed smear of bacterial culture, and it binds to peptidoglycans, the cell wall. So the bacteria start appearing as purple or blue to purple. And we don't know until now whether the bacteria are gram positive or gram negative. To strengthen the crystal violet, we add iodine, so the result will remain the same. After that, we have the most important step, which is the differential step, and we wash out the dye with acetone or ethanol only for seconds. Why? Because acetone or ethanol causes crystal violet iodine complex to dissolve away. So the more the time passes, the more the complex dissolve. And these few seconds needed to dissolve the complex in gram negative because they have much thinner peptidoglycans, whereas in gram positive because they have much thicker peptidoglycans cell wall, they need more than few seconds, maybe minutes, to dissolve the complex completely and to appear colorless as the gram negative. So don't prolong the procedure more than few seconds to stain the bacteria properly. Actually, we used to think that the third step or the differential step is the final and the last step in gram stain technique. Why? Because until now we have purple bacteria and we consider them as gram positive, and we have colorless bacteria and we consider them as gram negative. But actually, the purple ones are 100% gram positive, whereas the colorless ones are 50% gram negative. Why? Because we have a typical or variable bacteria that cannot stain well with gram stain, maybe due to the lack of the cell wall as mycoplasma, or they contains in their cell wall components rather than peptidoglycans as mycobacterium, or they are intracellular as chlamydia, and so on. So we should counter stain with safranin, or in some cases with thioxin, to stain the gram negative with the pink color and differentiate them from the remaining colorless atypical bacteria. Let's review the result of gram stain. We have gram positive bacteria, gram negative bacteria, and atypical bacteria. The gram-positive bacteria are violet to blue in color because they have a thick peptidoglycan cell wall that traps the crystal violet, whereas the gram-negative have a thin peptidoglycan cell wall that doesn't trap the crystal violet, but with the counter stain, they retain the safranin and appear as pink. Atypical bacteria. Those bacteria that don't gram stain well, so they appear colorless and considered as gram negative. The reasons: maybe they lack the cell wall as mycoplasma, ureaplasma, or maybe they have a typical cell wall. That means the cell wall consists of different components rather than peptidoglycans. For example, the mycobacteria have high lipid in percentage. The chlamydia lack the peptidoglycans. The treponema lack the lipopolysaccharide. Or maybe they have a very thin cell wall as leptospira. Or maybe they are intracellular ones as chlamydia, rickettsia, borrelia, anaplasma, bartonella, reliquia. To memorize all of these names of atypical bacteria, I have mnemonic here. These atypical microbes usually lack color because these microbes are bare, that means no cell wall, emaciated, thin, and remain inside. This refers to treponema, atypical anaplasma, microbes, mycobacteria, usually ureaplasma, lack leptospira, color chlamydia, because borrelia, microbes, mycoplasma, bare, bartonella, emaciated, reliquia, remain, rickettsia. The second consideration in the classification of bacteria is the morphology, and it comes along with the gram staining, and it divides into two main types: the cocci, which is the plural of coccus, and the bacilli, which is the plural of the bacillus. The gram-positive cocci are further divided according to their arrangement into clusters as staphylococci, pairs as streptococcus pneumoniae, enterococci, peptostreptococci, or chains as streptococci. The gram-positive bacilli are either aerobic or anaerobic. Clostridium actinomyces are anaerobic gram-positive bacilli, and both of them are spore-forming. Moving to gram-negative, they are either diplococci or bacilli. Diplococci as Neisseria, Marexella. Bacilli are further divided into Enterobacteriaceae as Escherichia coli, Cucubacilli as Haemophilus influenzae, other as Helicobacter pylori, and Vibrio cholerae. Normally, our immune cells as neutrophils try to protect our bodies from the invading microorganisms, including the bacteria, by creating the reactive oxygen species. They start converting oxygen into superoxide by NADPH oxidase. Then they convert the superoxide into hydrogen peroxide by superoxide dismutase enzyme. Then the hydrogen peroxide is further converted into hypochlorite, which is a strong microbiocidal substance that can kill the microorganisms, including the bacteria, as bleach. Unfortunately, some bacteria are sneaky and they can prevent this part of process and avoid being damaged by the hypochlorite, which is the microbiocidal substance, by having the catalase enzyme. The catalase is an enzyme that is responsible for the conversion of hydrogen peroxide, which is harmful to them, into water and oxygen, which are harmless to them, producing bubbles. Which microorganisms are catalase positive? First, put in your mind that catalase is not found in anaerobic bacteria because the formation of reactive oxygen species, including the hydrogen peroxide, needs oxygen. Actually, the catalase enzyme is found in many organisms as Staphylococci, Escherichia coli, Nocardia, Serratia, and so on. However, it is used predominantly to differentiate between the staphylococci, which are positive and capable to produce bubbles, from the streptococci, which are negative and not capable to produce bubbles. Clinically, patients with chronic granulomatous disease are deficient in NADPH oxidase enzyme, which is responsible for the conversion of oxygen into superoxide. So it contributes in the ROS formation. So those patients are experiencing recurrent infections with catalase positive organisms. Here we have coagulase enzyme, which is responsible for the conversion of fibrinogen, the soluble one, into fibrin, the insoluble one. By fibrin, the bacteria can surround the area of infection to protect them against phagocytosis and at the same time prevent the spread of infection and make it localized. This enzyme used predominantly to differentiate Staphylococcus species. Staphylococcus aureus bacteria are coagulase positive, result in clot formation. Most other Staphylococcus species are coagulase negative, result in no clot formation. Hemolysis test. Hemo, blood. Lysis, the breakdown of cells. Hemolysis, the breakdown of blood cells, the erythrocytes. And this test used to determine whether or not the bacteria have the ability to break down the erythrocytes and hemoglobin. And it serves to differentiate the streptococci based on the various types of hemoglobin degradation. This process is catalyzed by an enzyme called hemolysin, and there are several types of this enzyme, and each one has a specific pattern of hemolysis, and according to them, the streptococci bacteria are divided into three groups: alpha, beta, gamma hemolytic bacteria. Alpha hemolysis, in which there is a partial degradation of hemoglobin, and the remaining one is oxidized into methemoglobin, which is a substance with a characteristic biliverdin green color. Green hemolysis. The bacteria in this group are Streptococcus pneumoniae, verdens Streptococci. Beta hemolysis, in which there is a complete degradation of hemoglobin with a translucent halo around the bacterial colony. The bacteria of this group are Streptococcus pyogenes, Streptococcus eglactii. Gamma hemolysis, in which there is no induction of hemolysis. That means the agar around the colonies remains unchanged. The bacteria of this group are Streptococcus bovis and Enterococcus. Here we have another test other than hemolysis test to differentiate the streptococci or enterococci, and it is based on the presence of specific carbohydrates on the bacterial cell wall. And according to the presence of these specific carbohydrates, the bacteria or the streptococci are classified into three groups. Group A as Streptococcus pyogenes, Group B as Streptococcus egalactiae, Group D as Enterococcus and Streptococcus bovis, and the non-Lancefield bacteria, those bacteria which don't have these specific carbohydrates on their cell wall, as Streptococcus pneumoniae and Viridans streptococci. Now you're wondering why I didn't mention the group C. Actually, there are other groups as group C, E, F, G, and H, but fortunately they're less frequent with human diseases and they're not important unless we are veterinarians. What is the difference between Lancefield serotypes and hemolysis test? Actually, group A and group B equal beta hemolytic bacteria, group D equals gamma hemolytic bacteria, and the non-Lancefield bacteria equals alpha hemolysis. And here we have the note that we have mentioned before, that the Enterococcus, which were formerly known as group D Streptococcus, became a separate genus in the 1980s. Lactose fermentation, it is a major differentiating characteristic of gram-negative rods. It used to differentiate gram-negative rods, and it is usually tested by culturing unknown bacteria on McConkey agar. What are the components of McConkey agar and what are their functions? Agar, structural support for growth. Both bile salts and crystal violets inhibit growth of gram-positive bacteria. Lactose, nutrients for lactose fermenting bacteria result in acidic metabolic products, so the pH is gonna be reduced. Peptides, nutrients for non-lactose fermenting bacteria, result in alkaline metabolic products, so the pH is gonna be increased. Neutral red, dye which changes to a red color at low pH. So when the bacteria ferment lactose as a nutrient, the neutral red is gonna change into a red color. Here we have a mnemonic for this. Mac and cheese. McConkey agar is used for lactose fermenters. Here we have the classification of bacteria, particularly the gram-negative rods, according to lactose fermentation on McConkey agar. Lactose fermenters or lactose positive, those bacteria which can ferment the lactose rapidly, as Escherichia coli, Klebsiella, Enterobacter. These bacteria ferment the lactose in a rapid process, result in acidic metabolic products. So the neutral red is gonna change into a red color, as you can see here. But because of the rapid fermentation by this bacteria, there are halos or red halos around the colonies. By these red halos around the bacterial colonies, we can differentiate the lactose fermenters from lactose slow fermenters, as Citrobacter serratia, in which the bacteria can ferment the lactose, resulting in red or pink color, but without the red halos around the bacterial colonies. Lactose non-fermenters or lactose negative, those bacteria which are not capable to ferment the lactose, so the neutral red is not changed into a red color, and the bacterial colonies appear as translucent. The bacteria of this group are the gram-negative rods that we have not mentioned in lactose fermenters or lactose slow fermenters, as Pseudomonas, Proteus, H. influenzae, Salmonella, Shigella, Stenotrophomonas, and Acinetobacter.